All right, welcome to what I believe is actually podcast 47, which you are likely only watching if you are indeed signed up to take the AP exam, because we're not going to cover this content as part of the class. But I do want you to have some basic knowledge of bacteria if you're going to take the exam. You already have a considerable amount of knowledge of bacteria, given that we did the bacterial transformation lab, if you'll remember, with the P-Glow and the glow-in-the-dark gene that we inserted on the plasmid into bacteria to make them glow. We learned a lot about bacteria then, but I wanted to give you a basic overview. So let's get started so we can hopefully make this go as quickly as possible. First of all, when you hear bacteria, you need to think prokaryotic, okay? Please remember basic taxonomic information that you should have. There are two basic types of life on planet Earth: prokaryotic life and eukaryotic life. When we're talking about prokaryotic life forms, we are talking about bacteria. All bacteria are prokaryotic. What does that mean? Well, we'll talk about that some more in a minute, but remember, these are the most simple life forms, very little organization within the cell. There are no organelles, no nucleus, okay? Eukaryotes are, of course, have organelles, have an enclosed nucleus, and so on, and more complex. So let's put, if we do the traditional old school 5 kingdoms of life, bacteria are all off to themselves in one kingdom. That's since been revised, but let's think of it. They're all by themselves, and then eukaryotes make up the other kingdoms. Eukaryotes are everything that is not a bacteria. A protist, which is again a bad kingdom, but we'll it's kind of the catch-all kingdom for every eukaryotic organism that they couldn't put in some other kingdom. Fungi, plants, and animals are all in the eukaryotic group. So we are in this PowerPoint here, okay? Please remember that bacteria, prokaryotes, came first. These are the most simple life forms, the first life forms to evolve on planet Earth. Our very oldest fossils are of prokaryotes at around 3.8 billion years of age. So all life arose from prokaryotic organisms. When we classify prokaryotes, there are 2 major branches of prokaryotes, and we see bacteria, they all look the same to us, but fundamentally these 2 branches are so different from each other that the level of kingdom was not broad enough to distinguish them. Remember, we've got kingdom, phylum, class, order, family, genus, species, and kingdom Animalia, kingdom Plantae. These are pretty broad categories. Well, it wasn't broad enough to separate these 2 groups of bacteria, so they actually developed another level on top of the kingdom called the domain. We briefly mentioned this back when we were doing animal classification earlier in the year. They are so different, it was determined by looking at ribosomal RNA. If you'll remember ribosomes are made up of, they're a complex of globs of protein and something called rRNA, or ribosomal RNA. It's just a type of RNA. Messenger RNA, tRNA, well, there's rRNA. And ribosomes are globs of protein and RNA. By studying the nature of the ribosomes, they have found that these organisms are fundamentally very separate from each other, and also by looking simply at their genes, at their genomes, at their DNA. Okay? So the classification was revised. There is a universal ancestor for all domains: the regular bacteria, the archaea. These are also called eubacteria sometimes, but domain bacteria, domain archaea are 2 prokaryotic domains, and then the eukarya, which is all of the eukaryotic kingdoms. Universal ancestor to all, but if you'll note, based on lots of evidence that you see here looking at the nature of the nuclear envelope, the nature of organelles, the cell wall, lipids in the cell membrane, RNA polymerase makeup, blah, 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 blah. On down, presence of introns, remember that term. By comparing bacteria, archaea, and eukarya in terms of these fairly technical categories, it's been determined that in fact the archaea, the ancient bacterial group, is actually more closely related to eukaryotes than they are to other bacteria, and it is thought that we actually share a common ancestor with these guys more recently than we do with these guys. Okay, so just food for thought. So bottom line, eukarya and archaea share a common ancestor that lived more recently, just said this, than between the archaea and the bacteria. So I'm going to go back. We share a common ancestor with this group more recently than this group does with the bacteria, or than we do with the bacteria. So even though the archaea and the eubacteria are both prokaryotic, it is thought that in fact we are more closely related to the archaebacteria than they are to the eubacteria. We're going to talk about the what are archaebacteria and eubacteria. Archaebacteria, you should think archaeology, ancient. This is an ancient group, as are bacteria as well. But what you need to understand is they inhabit extreme environments. Most modern organisms cannot survive in these environments. These are the few that can. The environments in which these organisms survive are thought to be extreme, like perhaps the early Earth was extreme, hence the archaea name. We'll talk more about them later. The eubacteria are more numerous. Most bacteria are in this group, and again, sometimes they are just referred to as the bacteria. They are the ones we generally bump into. If we're going to be made sick, it's this group that's going to do it, not the archaebacteria. Now, all prokaryotes, general characteristics, whether it is archaea or eubacteria, they are extremely diverse, extremely numerous, and extremely diverse. Every type of metabolism, meaning sets of chemical reactions that allow us to consume nutrients and create energy and all that, every type of metabolism that has ever been on Earth has been created by prokaryotes. Of course, prokaryotes are unicellular. The cells are extremely tiny. This is the end of a needle, like the pointy end of a needle, and these are bacteria that are on that pointy end of the needle, obviously an electron micrograph. This is why we sterilize a needle before we use it to take out a splinter. So very tiny, much smaller than eukaryotic cells. They do have cell walls. They do not have compartmentalization. Remember, no compartments, no organelles, no Golgi apparatus, no ER, no nucleus, things like that. Some bacteria are capable of movement, some are not. Speaking of movement, let me talk with you a brief bit about some structures in bacteria and those in eukaryotes. Eukaryotes and bacteria have similar structures. We will find in prokaryotes and eukaryotes cell walls, for example. Plants have cell walls. Bacteria have cell walls. But these are not homologous in terms of evolution. These structures do not indicate common origin, because even though they look similar to us, they are fundamentally different. They are analogous structures. In other words, in bacteria, the cell walls are made out of a substance called peptidoglycan, which is fundamentally different from cellulose, which is what makes up plant cell walls. Flagella in bacteria are analogous to flagella in eukaryotes. We both have flagella, or structures that look like these whip-like structures that will propel a cell, for example, a sperm, along in a viscous fluid. But the mechanism by which the flagella move in a bacteria is fundamentally different from the way they move in eukaryotes. So superficially similar features, cell walls in bacteria and eukaryotes, or flagella in each, but they are fundamentally different. They evolved independent of each other, not one descended from the other. Okay, so not evidence for common ancestry there. This is a eukaryotic flagellum, just to remind you. It is made up of microtubules that are in little bundles, 9+2 arrangement that go around, 9 bundles of microtubules. Remember, microtubules are fibers that extend through, that do many things, and mitosis and so on. Well, microtubules are in the flagella as well, and there's a bundle of 2 in the middle. So 9 bundles around and 1 bundle of 2 in the middle. And there are little motor molecules, little proteins that connect these little rods, and they make them slide past each other so that the flagellum ends up going back and forth like this. In a prokaryote, the mechanism is fundamentally different. There's almost a cranking type mechanism, and so what happens is the flagellum whips around in a circle. It is a fundamentally different mechanism. That is why these structures are analogous to each other and not homologous. Okay. Also, please remember, this is a good time to remember the word taxis. We did this in our animal behavior unit. Remember, if an organism exhibits taxis, it moves toward or away from a stimulus. Kinesis is it's just kind of moving randomly. Taxis, we have a directional movement. Bacteria can exhibit taxis as well when they move. Now, internal structures of prokaryotes, remember, no nucleus. They do have a nucleoid region where you would find the DNA, but it's not encased in a membrane. There's no membrane-bound organelles, no compartments. However, there are some, and this diagram doesn't fold it, but there are some foldings of the cell membrane that can enfold like that, and there's various chemicals embedded in the membrane that can do certain processes, but there's no compartmentalization. But there can be some specialized activity in the cells of prokaryotes because of those foldings. The genome, remember, is fundamentally different from ours. It is a single circular ring of DNA, so like that, but it's still a double helix. I'm going to try to make this look like a double helix ring. Good luck. But it is just one circular chromosome. You can see it's folded all over on itself, but we do not have linear chromosomes like we are used to in eukaryotes. We do not have that, and hence we don't have mitosis. Mitosis occurs with linear chromosomes. Bacteria don't do mitosis. Remember also about plasmids, we have little small rings of DNA that carry often accessory genes, like perhaps genes for antibiotic resistance, like we learned about with our pGlow and the ampicillin resistance and so on. So they're little accessory molecules of DNA. Prokaryotes do have ribosomes because just like any other cell, they have to make protein, and so ribosomes are a very ancient type of structure, and bacteria do have ribosomes. They do have some significant structural differences from our ribosomes, but nonetheless they are ribosomes. Okay? Reproduction in bacteria is asexual. If you are a bacteria, you will be having no sex. Okay, you will reproduce asexually by means of called binary fission, not mitosis. No mitosis. The ringed chromosome is fundamentally different. Binary fission, the genetic material copies itself, as you can see here. Then the cell divides, and boom, that's binary fission. It is quick, 20 minutes in some cases from one generation to the next. Pretty much the only thing that limits bacterial growth is some limiting nutrient, a limiting factor, if you will, in the environment that is going to limit the amount they can grow. So essentially, they clone each other. Now, that said, we know that organisms need to have variety among their population too, or they will often die out if they bump into unfavorable conditions. We need to have some variety there. Variation does occur, even though reproduction is asexual. Most variation occurs via mutation, okay, mutations in the DNA, but it can also occur from just reshuffling genes. But wait a minute. Sexual reproduction is the fundamental way we do genetic reshuffling or recombination, and I just told you there's no sex. So how can we have recombination with no sex? Okay, how can we reshuffle the genes if we're not going to have sex? Well, remember we talked about with our lab bacterial transformation, where a bacteria picks up a stray plasmid. They can also pick up stray fragments of other types of DNA from other bacteria, but we talked about it in terms of plasmids. They pick up plasmids from their environment. Boom, you've got some variation. Conjugation is when two bacteria hook up, they connect their cytoplasm and exchange a plasmid or some other bit of DNA. It's not sex. It kind of looks like sex, some weird's going on there, but it's not sex. Sex is an equal contribution from two parents towards an offspring. This is not an equal transfer. One organism is maybe picking up a plasmid from another. It's not sex. Transduction is something we have not talked about. Let me get rid of this. Transduction is, ignore this over here and this over here. Transduction is when a virus has infected one bacterium and then it's picked up some DNA from that bacterium, and then it goes and infects another and shares some genes from this bacterium with this virus. So this is a viral transmission of genetic material. Okay, kind of odd. Usually I don't think you'd be asked about that, but it does occur. Okay. Please remember, none of these is sex, not an equal contribution from two partners. We're basically picking up in all cases pieces of genetic material, not full complements of genetic material. Remember, of all of those, mutation is the major source of variation. Because bacteria reproduce so quick, if you get a favorable mutation, it can be really quickly passed to a large number of offspring in a very short period of time if it's favorable. So adaptation to the environment can be very, very rapid, which explains a lot about bacteria becoming resistant to antibiotics. If a mutation pops up that makes a bacteria resistant, then boom, it's spread, and the next thing you know, your antibiotic's not killing that bacteria anymore. Okay? Alright. Metabolism. Obtaining, using nutrients and energy. The most famous metabolic processes we know of are cell respiration and photosynthesis, things like that. Every type of metabolism that exists on Earth first evolved in prokaryotic life. So again, we owe prokaryotes a whole lot. Some fundamental types of metabolism that evolved in prokaryotes: photoautotrophism, or photosynthesis. Photo means using light, autotroph to make your own food, so we're talking about photosynthesizers. Chemoautotrophs, they make their own food too, but they don't need light to do it. They use certain inorganic compounds to give them the energy to make their own food. We'll talk more about that in a moment. And then chemoheterotrophs, or we would probably call them just plain old heterotrophs, like us. We're heterotrophs. We eat stuff to get the energy we need. They consume organic compounds. They eat stuff to get the energy that they need. Okay, and then there's other weird metabolic forms as well. We won't speak of those. But please do remember that photoautotrophs and this is true for any photoautotroph, a plant or whatever, but in terms of bacteria, they do photosynthesis. Now, bacteria are different from plants in that they do not have chloroplasts. Remember, there's no organelles in bacteria. The photosynthetic pigments that we would find in chloroplasts in plants are actually just located in the cell membranes of photosynthetic bacteria. They can photosynthesize without the organelles themselves because the chemicals they need are embedded in their cell membranes. Cyanobacteria are the most famous type, so this is an example of a photoautotroph, and there's a picture of some there. And it is thought that cyanobacteria, or something like them, cyano means blue-green, they do look kind of blue-green because of their photosynthetic pigments. These guys are the guys initially responsible for putting oxygen into Earth's atmosphere at all. When Earth first formed, there was no oxygen, but these life forms doing photosynthetic activities for the first time, remember we have CO2 plus H2O plus light is going to give us sugar plus oxygen. Boom. All these guys started releasing this into the atmosphere, and after a while the atmosphere has a surplus of oxygen, so we owe these guys a lot. Without them, our life form couldn't exist. Chemoautotrophs, similar to photoautotrophs because they do make their own food, but they don't need sunlight to do it. They use inorganic compounds like maybe nitrogen, some chemical, as their energy source to make their own food. In other words, these guys can be primary producers just like photosynthesizers can, but they don't need light to do it. At the deep sea vents, there are bacteria that are using the chemicals that come out of the deep sea vents to make their own food, and then whole ecosystems, these worms, for example, rise up around them. Okay, so in this case, these bacteria are the primary producers in that environment. Chemoautotrophs are exclusively prokaryotic. We don't have any eukaryotic ones. They're also really important in our ecosystems above the deep sea vents as in nutrient cycling. For example, nitrogen fixing bacteria are really important in cycling nitrogen through our ecosystem, and we talked about that with ecology. We'll review it some as well. Chemoheterotrophs, or just plain old heterotrophs like us, eat stuff to get energy. Most prokaryotes are in this group, and they have a whole ton of important ecological roles. They can be saprobes, which are essentially, it's just another fancy word for a decomposer, and Lord knows we need decomposers to break down dead stuff and return nutrients to the nutrient cycles. They can also be parasitic, and you've probably had a relationship with a parasitic chemoheterotroph that gave you a bacterial infection, and that's not fun. Alright, bacteria and oxygen, just real quick. Some bacteria require oxygen because they do cell respiration. They're called aerobic. They are called obligate. If you're obligated, you have to. So obligate aerobic organisms. Some bacteria do not require oxygen, and in fact can't be in the presence of it because oxygen can be very reactive and caustic. These are kind of ancient bacteria, or throwbacks to ancient bacteria, that are anaerobic bacteria that cannot live in the presence of oxygen. They do fermentation instead, and some can go either way. These are generally called facultative aerobes, meaning they can use oxygen or not. Doesn't really matter to them. They can go either way. Some important evolutionary events around prokaryotes, really important for all of us. Glycolysis, the first part of the cell respiration biochemical pathway, evolved in prokaryotes. Photosynthesis evolved in prokaryotes. Chemiosmosis, the whole ATP synthase idea, using a proton gradient to make ATP, evolved in prokaryotes. Hugely important, major watershed evolutionary events for life on Earth all happened within prokaryotes first. Now, this from here on is a very quick look at the different groups of prokaryotes. Again, domain Archaea, these are the extremophiles. Think of these as living in that harsh early Earth environment when there was no oxygen. It wasn't like we know it here. There are 3 types we'll mention. Methanogens, they are anaerobic. Basically, they use hydrogen gas to reduce CO2, kind of like, in other words, to kind of make a food item out of it. If you smell marsh mud and it smells funny, these guys are what's causing it, because they're releasing methane gas, which is a smelly gas. It smells like sulfur. They could act as decomposers for sewage treatment plants. They even help cows digest their food. You know, a cow has many complex stomachs with bacteria in there helping them break down the cellulose. These guys work in doing that as well. Halophiles tolerate extreme salt. Some actually require it, some just tolerate it, but extreme salt conditions, like in the Great Salt Lake, you might find some of those. Thermophiles tolerate extremely hot environments, like sulfur springs, deep sea vents, things like that. Now, domain bacteria is where most prokaryotes are. Now here's a lot of funky names. We did learn about these. These are the ones that are photosynthetic. They're in domain bacteria. Okay. This is showing you kind of a more detailed view of the family tree of all these guys. Here's us, we eukaryotes, and here we are being more closely related actually to the archaea than to the bacteria. And here you can see there's very many more groups of domain bacteria than there are over here. Okay. Last thing we'll talk about is the impact of prokaryotes on our world. Okay, they are of course very important in the biogeochemical cycles, recycling of nutrients. They act as decomposers. They act as photosynthesizers. They act as nitrogen-fixing bacteria, so really, really important in our ecological cycles. They can act in symbiotic relationships. Legumes, bean-type plants are in a symbiotic relationship with nitrogen-fixing bacteria, again helping our nutrient cycles. We are in a symbiotic relationship with E. coli, not the kind that makes you sick, but there's a kind that lives in your gut and helps you. There's a particular bacteria that helps you get vitamin K. You can't get it any other way. So we are engaged in symbiotic relationships with E. coli in our gut all the time. If you don't have E. coli in your gut, you usually have pretty serious problems. Of course, prokaryotes, bacteria also give us disease, usually caused by toxins they secrete, not because the bacteria themselves are doing anything to you, but because they secrete toxins that make you sick. And they can really cause nasty problems if your immune system can't fight them. They can kind of grow out of control and produce too many toxins. Okay? And of course, prokaryotes are used in industry all the time. E. coli is the white rat of the laboratory setting. We experiment on it all the time. They decompose things for us. Even the big oil spills, we use bacteria to decompose the oil that gets spilled periodically. We use them to produce antibiotics. We use them in food to make cheeses and stuff. And of course, we use them to make gene products that we want. Human insulin, like with the Piglo experiment, if we had inserted the gene for human insulin instead, we could have had bacteria making human insulin. So they do a lot for us. And that's it. And I know that went fast but long. How can it be both? I hope you got something out of it, and let me know if you got any questions. See you next time.